just to emphasize, my quiz is worth 10 points, 20 multiple choice questions. Pay attention to what we talked about here and read those small articles, and you've got yourself an easy 10 points. 10%. Sorry, John, don't talk in points, percentages. Okay, up to now then, hopefully you're still following this part of the course. We've been talking mainly about biotechnology, the advances in science, particularly in microbiology, and how it's led to decisions we have to make in the broad arena of bioethics. And we've really focused on manipulations of organisms, going in and changing the gene structure or making those organisms manufacture a product that we'd like to use. But I want people to be really clear that bioethics extends way beyond just those kind of biotechnological innovations. And I want to start today by giving you a case study of a real event that happened over the last 5 years and was being considered as a fairly dramatic ecological event. And then, of course, as the reality came home, then the public and the science community started to worry about it, and of course then we had to think about it ethically. So this is a kind of case study, a true story, and after that, if we've got time, I just want to go on to sort of some other examples of biotechnology that don't involve going in and manipulating the organism, but are fun examples of microbiology in the hope that some of you might really get interested and become microbiologists. So let's start with this broader view, this ethical challenge in science, and I'm calling it environmental meddling. And to understand this, we have to again give you a little bit of background relevant to the subject area. So sort of bear with me. Hopefully you're looking at these two micrographs. Let me make it bigger for you. These two micrographs, and everyone's saying to themselves, I know what these are. These are algae. Yes, they're green in color, they're single cells, and therefore they're examples of these kind of microbes we refer to as algae because they can photosynthesize. They can use sunlight to make new biomass. And of course, we've shown one of them before, and you better be saying to yourself, that's a diatom, good. This one here is called a dinoflagellate, just two examples. And these organisms, when they're out there in nature, when the conditions are right, they can form explosions of the population, and we've mentioned that already. These sudden proliferations of the population give rise to what's called a bloom, an algal bloom, and that is so dense that it discolors the water and you can actually see it. And you walk across campus, some days the pond is a fairly green color, and that's because of this algal bloom event that's in there, which we call a eutrophication event. So our pond has never looked like that, thankfully, but if it did, that would be a very strong algal bloom and a clear case of eutrophication. And these areas are really quite problematic around the world. They obviously make the pond here somewhat toxic because what happens is there's so much biomass there, so much explosion of green stuff that at some point it dies. It can't keep on growing forever, and it dies, and the bacteria there, as you probably know, are the organisms which will degrade and break down material, recycle it. And so there's so much explosion of bacteria then after this event that all the water becomes what's called anoxic. All the oxygen is stripped out of it. So anything that's living in that pond that relies on breathing, and that would be all the animals, all the fish, they all die. And so it's a very serious event, particularly when it happens in a fairly large scale. Every year now, we have a Gulf dead zone develops, and that's because of all the fertilizers coming down the Mississippi River, because of all the intense farming all the way along the banks of the Mississippi, and all this fertilizer rich in nitrates and phosphates causes all the algae to grow, and they get so dense and die and suffocate the water that all the shellfish in this area are being killed off. And this isn't the true color, this is taken as a satellite image, but this red color is the area that's known as the dead zone. And this is happening every year now in the Gulf, and it's causing incredible hardship to the fishermen who rely on these shellfish for their livelihood. The other kind of algal bloom that you should be aware of, and hopefully you already have heard about because you just have to read a newspaper and you can't avoid them. Sometimes these blooms develop and they have a different color to them. They're made by green organisms, but these organisms can also have accessory pigments, different pigments inside them, and often they take on a red color. And so we hear about these red algal blooms, or red tides as they're often called. So just be clear though, those are explosions of those microbes, in this case usually dead dinoflagellates. So this gives rise to a slightly different problem, because many of these red tides, not only do they strip out all the oxygen in the same way that any algal bloom does, but some of these dinoflagellates, these red pigmented ones, give out toxins, and that toxin then gets into the food chain. And on day one, seems like a long time ago when I was lecturing, we talked about this toxin that's particularly nasty if it gets into shellfish, because the shellfish are filter feeders and they're eating these algae and they're taking the toxin into the tissues, and then if you eat them you can get poisoned. And so we have to watch out, particularly for one called paralytic shellfish poisoning. That's the most severe, but there are many of them out there. And it's caused by eating contaminated shellfish that have been feeding in an area where there is a red tide. The particular toxin that causes paralytic shellfish poisoning is known as saxitoxin. And just recently, well, saxitoxin, stick with that one for a moment. That is the most devastating one. I was being serious in the first lecture when I said if you ever feel a tingling when you're eating shellfish like a mussel or shrimp, then stop eating it, because you could be coming in contact with saxitoxin. If you were to keep eating saxitoxin, it does paralyze you. It paralyzes your breathing apparatus, your diaphragm, and you effectively suffocate to death. There is no cure for it, and we lose about 5 to 10 people a year across the globe, so it's not something to fear, particularly in America where we're very aware of this problem, and there's a lot of regulation to make sure shellfish never comes in contact with it, but it's still something that could happen, so be aware of it. And it's not just dinoflagellates that cause it. There was a case in Prince Edward Island in Canada 10 years ago where some people got very sick from eating shellfish. Some died, many were hospitalized. It was a completely new toxin that we knew nothing about, which turned out to be called domain acid. And it turned out it was actually from a diatom, not a dinoflagellate. But again, same problem. We have to be very careful about these algal blooms. Okay, this is background information. I'm giving you the pieces to understand the problem that we were facing when we came up with this ethical dilemma that came about just a few years ago. The next piece of background information is photosynthesis is the process that makes biomass on this planet, as you know. And it occurs by dragging CO2 into the organism, be it a plant or be it a green microbe. The CO2 is converted to biomass. That's what the process of photosynthesis is. Now, oceans cover most of the planet. In case you didn't know, 70% of the planet is made up of the oceans. And so, because the oceans are all bathed in sunlight, at least to a depth of perhaps, depending where the ocean is, 50 meters, there's a lot of area there on which phytoplankton, these green algae, these diatoms, these dinoflagellates, can photosynthesize and grow. So much of it is happening there that it's been called perhaps the most important process for life on Earth is the reduction of CO2 into green things. And we think up to 40% of global carbon assimilation is in the oceans. Okay, that might be a little complex for those of you that are not biologists. We're just saying that while you think about most of the photosynthesis happening in rainforests and fields and all the green plants you see, up to 40% of global photosynthesis is occurring in the oceans. Hugely important areas for turning CO2 into biomass. Okay, we know that global climate change is happening. I hope we're all on the same page with that. And there is climate change happening now, and you've all heard about greenhouse gases, and that's why the planet is warming up. And we know that CO2 levels are rising because we've been monitoring them ever since the 1950s. So it's an irrefutable fact that the amount of carbon dioxide in the atmosphere is increasing. And that graph shows it increasing just since the 1950s. They monitor it every year. And there's a dramatic increase, and it's still increasing because of all the industrialization, all the cars. You know the story. The consequence of this blanket of CO2, of course, is the planet is warming up and climate is changing. We also have great data that I haven't shown here, but you can go further back in time because you can actually analyze little pockets of gas that you can find in ice in the Arctic and Antarctic, and those trapped gases take you all the way back to the 1800s. So we can actually see when CO2 in the atmosphere started to dramatically increase, it was because of the Industrial Revolution and everything we've done thereafter. Okay. So wouldn't it be nice if all these algae in the ocean could save the planet? Think about it. If they were all to drag in that extra CO2 that's coming into the atmosphere, they could remove it. And that's what phytoplankton do. They grow by taking in carbon dioxide. Now that doesn't help you much if they die and they're broken down by bacteria, because then the CO2 just goes back in again. But what would happen if they take it in, they die, and they sink very fast, so that they were actually buried in sediment 8,000 meters deep in the ocean and got buried before they got broken down to release their carbon dioxide? That's a subject that became the focus of every oceanographer in the world, basically, about 10 years ago, trying to understand what happened to the phytoplankton that grew and how quickly they sank and what happened to them when they get buried. And the feeling was that if they do sink fast enough, then they can get buried in sediment and effectively remove carbon dioxide from the planet for a good 4,000 to 5,000 years. Wow, that could be nice. That subject is kind of summarized in those three statements. As CO2 increases, more is dissolved in the water, and more is then taken up by the green things in the water. These are called the phytoplankton, or algae, call them what you like. They take up the CO2, and if they sink fast, they get buried in sediment. And that is known as biological pumping. So, this got a lot of people interested, because the next little piece of the background information was that there are all these areas of the ocean that have got lots of light, lots of sunlight, lots of big nutrients like nitrates and phosphates, which are the two major nutrients. Just like your tomatoes at home, you want to grow them well, you put on nitrates and phosphates. There were lots of areas of the ocean that should have been really good for growing algae of the kind we want to grow, drag out the CO2 and sink. But they had very few algae actually growing there. In other words, if you were to look this up and Google it, these initials have been used for the last 10 years. They're on the tip of every oceanographer's tongue. A high nutrient, low chlorophyll area, HNLC areas. Don't be confused by that. High nutrient means lots of nitrates and phosphates, things that would normally grow plants. Low chlorophyll, chlorophyll is the pigment that makes a plant or an alga green. It just means that there's not very many algae there or not many phytoplankton there. So these were confusing areas of the ocean which should have had a lot of algae growing in them that could be stripping out the CO2, but turned out that they had very low amounts of algae growing there. So a lot of people and a lot of money went into trying to answer, what is the problem here? And can we stimulate these algae to grow better and save the planet? And the trace element that scientists zeroed in on was iron. Iron is one of the components that actually makes up the pigment chlorophyll. It's a little bit like you need iron to make blood cells. It's a clumsy analogy, perhaps. But you don't need very much of it. And algae don't need very much of it either, but they do need it to make their green pigment, just like we need traces of iron to make our blood cells. And so the suggestion was by some scientists that, hey, this is easy. All we have to do is fertilize the ocean with iron. And it was calculated that if we added 300,000 tons of iron into the ocean, we could remove 2 billion tons of carbon dioxide. Wow, saving the planet. Sounds very nice. This was so attractive that there was funding made available, and they did an experiment off the Galapagos Islands, which was called the Iron X1 experiment. It was done in a large expanse of ocean, about 8 by 8 kilometers. And they added a modest amount, 455 kilograms of iron. And they then monitored that piece of ocean to see what happened. And yeah, it did. It worked. The algae grew in that ocean. They had fertilized it. The rate of photosynthesis went up, and the amount of carbon dioxide got stripped out of the oceans. This took about 10 years up until fairly recently of intense scientific work, lots of studies by many, many scientists. This was really a big deal because there was a feeling here that we had a way of solving the problem of increasing carbon dioxide levels and therefore climate change. And then came along a company headed by someone called Michael Markles. He was a scientist, and he decided he was going to make money at this. And he got a lot of attention because there was a lot of political interest in this scheme. And his plan was, I'm going to fertilize the Gulf Stream, which is the water body that flows up the east coast of America. I'm going to do it by adding 25 tons of iron. And this is going to be great news for the planet because it will increase fish yields. These phytoplankton, these green cells, these algae and diatoms are what fish eat. So if we can make them grow, then we will have more fish food, and therefore we'll have greater yields of fish to feed America and the world perhaps. And we will also be taking out all this CO2 from the atmosphere, and we will be solving climate change. And it got very close to happening until some scientists, particularly headed out of MIT by one of the microbiologists, Susan Chisholm. You don't need to remember that name, but MIT scientists got very nervous about this and weighed in. What could go wrong? You've got all the pieces of information from the background. Now this is the little piece of critical thinking. What could go wrong with this idea of Michael Markles to pour in iron into the Gulf Stream, give us more food, and solve climate change? Anyone that shouts it out gets an instant 100%. What might happen? Just based on that background information I just gave you. We might end up with an algal bloom. Think about it. We could pour all that iron into the Gulf Stream, generate a toxic algal bloom of a magnitude we've never seen on the planet before, kill all the fish, and cause irreparable damage to the ecosystem. And that is what suddenly stopped this madness. And when MIT biologists weighed in, of course you could stimulate the growth of a wrong alga. That was something that was never considered by industry. If it gone anoxic, you would release all this methane gas into the atmosphere. That's a byproduct of the breakdown of bacterial decomposition of algae. You might, of course, produce a toxic algal bloom, which would be even more damaging. But the methane gas alone would have effectively made the whole climate change problem worse, because some of you probably know that methane is actually 21 times more effective at trapping heat than CO2 in the atmosphere. Methane is a very problematic gas in terms of this blanket of gases that we're putting up into the atmosphere. So, clearly that was a very wise ethical decision to stop this experiment, but I need you to be aware that this was a 10-year, multi-billion-dollar experiment that very nearly came to completion off the east coast of America. We need people who are trained scientists who can weigh in and make informed decisions. Okay, sorry to disappoint everyone. We got this working, and but the good news is you can just pay attention and have fun with it and just let it reinforce some of what we've been saying about some of the microbiology. So I'm being serious from now till 9:30 or whenever you're dismissed. Just enjoy this bit. You don't need to pay too much attention. But it does sort of reflect things I've done in my life as a microbiologist, and I tend to focus on the ones which have got some technological aspect to them, which is what this course is all about. It all started for me when I was, I don't know what age, 26, when I got my PhD at the University of Stirling in Scotland, which is a lovely campus should you ever want to do a PhD abroad, situated around this lake. And I think I told you, or John told you when he was doing my bio, that I used to work on a type of organism, microbe, called a naked amoeba. And these are just some pictures of naked amoebae. I'm not going to talk about the kind of work I did there because it wasn't applied. It was straight microbial ecology, and I really want to focus on the kinds of research I've done in various places that have got some practical relevance or technological relevance. And so after I got my PhD in 1979, I decided Britain was too cold and wet, and I went to Canada, and I worked at the University of Toronto for 4 years. There I worked on oil-degrading bacteria, how to clean up oil spills, and in particular the research was looking at, is it important to have these consumers around which are eating the oil-degrading bacteria? So the question then is, if we want to help break down an oil spill, is it important to keep the bacteria there plus everything else, including those kind of microbial consumers? And these are examples of ciliates that we talked about in day one of this class. And you can do an experiment where you can break down oil. You can break down oil with bacteria, you can break down oil with grazers like these ciliates present, and you use an instrument called a gas chromatograph. And oil, as you probably know, is made up of lots of different hydrocarbons, all different carbon chain lengths, and that's what these peaks indicate. And you can then look at oil that's broken down or not broken down. But I don't want to get too technical, and so we could just look at it in pictorial form. And these are just the oil films on 3 flasks that were in the laboratory. So this flask, or the oil in this glass beaker or flask, shows control, meaning the oil doesn't have any bacteria with it. It's just sitting there. Some of it breaks down because it's evaporating, but there's no microbial breakdown of that oil. This second flask, B, shows a layer of oil that's got oil-degrading bacteria growing in it. And this particular flask did break down the oil more, although you can't see it because you needed that analysis with the gas chromatograph. But look at this one. Look at the oil changed there. That's exactly the same oil-degrading bacteria, but it's got these grazers present. And what's happened in this flask is that the breakdown of oil has been much more dramatic, and we can see that because it's broken the oil into little droplets, and that's a process called emulsification. So it turns out that having grazers present is very important. I won't labor why it's important, but just think about a bacteria that's being consumed by a consumer. It's then giving out waste products. And the waste that's coming out of that ciliate, that little grazer, is rich in nutrients, nitrates and phosphates and ammonia and other nutrients, which then stimulate the bacteria to grow better. And so what's happening here is a little recycling of nutrients in this bottom flask, where the bacteria are being nourished by what is being released by the ciliates. So this went on, and we looked at the effect of oil dispersants to see if they were toxic, and they are. You can put in oil dispersants to remove the oil, and that actually is incredibly toxic to all the ciliates, and so therefore by adding this mixture of chemical dispersants, you are actually preventing microbial breakdown because the bacteria are not being grazed by the ciliates and other grazers, and therefore they're not doing a good job. That's got relevance in the Gulf spill recently. If any of you were paying attention to that massive oil spill in the Gulf, what did they pour on the oil? Correct. Chemical dispersants. The name of it was Corexit, and that was one we tested way back in 1979, and it was shown to be incredibly toxic. And what did they do in the Gulf? They covered it in Corexit. Fascinating. That's all those years later. They didn't learn from research done in the 70s. I'm skipping through things because we haven't got that much time. After Toronto, I went to Nova Scotia for a while. We didn't worry about that. I then went back to England and worked at a government laboratory where I studied mainly the ecology of amoebae. That was a place called Windermere for anyone that's been to the Lake District in England. Nice place to work, except it's probably the wettest place in the whole of the British Isles. But anyway, there I worked on other naked amoebae, what they looked like, and did a lot of counting of them because no one else had paid attention to this numerical group of microbes that were just an overlooked group. So I probably spent about 5 years of my life publicizing just how common they are. No matter where you look, you can find many thousands of them in just a liter of any water from anywhere you go. I spent a lot of time getting into the business of identification of these types of amoebae, and it's worth just dwelling on how we identify microbes for a moment or two. Most of the microbes on the planet, as you've been told many times by me, are the bacteria. And this is quite an exciting area because we've always assumed we could identify bacteria by growing them up on agar plates. And so you take some water, you spread it in a thin layer on a nutritious agar plate, And the bacteria are really growing on the nutrients in the very thin water film that sits on top of the agar. Anyone that's done any microbiology will know what I'm talking about. So the bacteria don't actually eat the agar, it's like a sponge. They're just on that very thin film of nutrient-rich water on the surface. And so if a bacterium is to land on that surface, it divides and divides and divides. And I've told you how fast they divide. They can form billions of cells in the matter of days. And when they make billions, you can see them as a visible colony. So that yellow colony there came from one bacterium. Whether it's in a pond or an ocean, cannot be grown in the laboratory. We now know, because of molecular biology, that 99.9% of all the bacteria on this planet have never been seen in laboratory culture. This is huge for anyone that's in the field of microbiology, because everything we thought we understood about microbes is wrong, because we were using the wrong technique. So it turns out that the commonest bacteria in the ocean is something called SAR. It's not a very nice name. It's been found by molecular methods, but no one has ever seen it growing in a laboratory. And yet every water sample from the ocean contains it. So think about it. It's huge. It's really turned our understanding of microbial ecology on its head. And it underscores why we have to be very careful when we go in and manipulate nature, because we do not understand it. Just in the last 5 years, we've decided that all the microbiology done in the last 50 years is wrong. I went to California at that point. I got fed up getting wet in Windermere. It's a really miserable place to be, except when the sun shines. So I went to the University of California at Santa Barbara because someone had found an amazing amoeba that they wanted me to come and work on. So I went across and worked on this one called Trichosphaerium. This is a beautiful amoeba, because it's easy to identify. You can instantly recognize it. It's quite big. It's got these little strange projections called dactylopodia coming out of it. And although it looks naked, it looks like it doesn't have a coat on it, a shell. When you look at it with an electron microscope, you can actually see that it's got a covering. And those little projections came out of this pore that is on this shell. It's a very strange amoeba indeed. Sometimes when you grow it, it doesn't have this smooth covering on it. It covers itself in spicules. These are made of calcium carbonate. Amazing. This is why you want to study microbiology, you guys. It's so much fun. You're always finding something new. But the research group in Santa Barbara weren't too interested in what this thing looked like. They were interested because it made a very interesting enzyme. And there's great interest in this food crop. I know it doesn't look very nutritious, but this is a type of seaweed, which is known as porphyra. But if you go to buy it, it's called nori. And this is what you make those sushi out of. This is that covering that you wrap up the rice balls and the shrimp and everything else that you're putting into your sushi. This is flat sheets of this alga called porphyra. And we may not appreciate just how big a cash crop this one is in the world, because we don't live in Japan, in Asia, because over in Japan, they grow this in huge aquaculture areas around the coastline of Japan. And they do that because this is part of the staple diet of Asians. And there's huge interest in this. The problem with porphyra is this type of seaweed, it only grows for half the year looking like this. For the other half of the year, it goes into a microscopic form that just goes in and settles actually settles in shells and other areas in the ocean. And then 6 months later, it grows and comes out as porphyra. So that's a real problem, because if you're trying to make a cash crop and you're trying to sell this and make lots of money and satisfy the needs of the people, you want to be able to grow this 12 months out the year. And of course that then requires some manipulation of the alga, some crop enhancement, just like we've done with land crops, with wheat and barley. We've increased the yield by breeding them and genetically modifying them. We want to do that with porphyra. The problem is, to get any manipulation going, you have to get rid of the cell wall. This cell wall, this brown cell wall, it's actually red because this is a red alga, is very difficult to remove. It's called a phycocolloid. It's not like cellulose in a land plant, which is easy to get rid of because you just use cellulase and off falls the wall and out come the cells. No one has managed to get porphyra to break down the wall and release individual cells, which you then manipulate, fuse, genetically modify. You can then go to work improving things. So they desperately needed an enzyme that would take off the cell wall of this material. Seaweed, by the way, gives us very useful products. Some of you have heard of agar. We just mentioned it. That comes from the cell walls of algae. Another one is alginate. You've probably seen that. Carrageenan is another one. These are all products from algae, macroalgae like these seaweeds. And so there's a huge business out there in all of those products if we can manipulate and grow them better. Look at the packaging next time and see what you're eating. Ice cream, cheese, almost everything you eat is going to have carrageenan or alginates in it. Next time you have a McDonald's cheeseburger, that cheese isn't cheese. It's alginate and carrageenan from seaweed with some orange coloring in it. Lovely. Yum, yum. Won't do any harm, but don't be fooled to think it's cheese. So these are important cash crops. And so the research group in Santa Barbara had found that this amoeba can break down seaweed walls. So if you take a piece of seaweed and put it in a petri dish, this entire area used to be a blade of porphyra. All of this digestion is due to enzymes breaking down this seaweed. So I didn't take that project any further, but that research is still going on in Santa Barbara. Checking the time. I decided I wanted to go work at the University of London, so I went to their marine station, which is on an island off the coast of Scotland. But for 8 years, I was faculty at the University of London, based here at this marine station. Oh, that, by the way, is marine snow. Marine snow is the name of all these dead pieces of phytoplankton that stick together when they die and then they sink. Huge interest in how fast these marine snow particles sink, because that, remember, is all about whether these dead phytoplankton can remove CO2 fast enough to do biological pumping. So I did quite a lot of work on that piece of the puzzle that we described as the case study earlier. Can these organisms really remove CO2 from the atmosphere? I had some great times there because I got to work with John Watson at the University of Aberdeen. He's a physicist, and he had found a way to take holograms of all the particles in a tank. And that was his tank in his laboratory at the University of Aberdeen. And being a physicist, he knew nothing about microbiology. And so I got a call one day from him, and he said, "Can you bring up some microbes so that we can see if we can spot them with this hologram?" And I got quite involved in this research group. And for the life of me, I cannot explain to you what a hologram is and how it works and how it's taken. But this lab had a series of lenses and lasers that would fly around the room and go all through turns and twists and suddenly capture the particles in this tank within a 50,000th of a second, which was the speed of the ruby laser. And he could then replay the hologram and see where every little particle was in that tank. Pretty exciting, and you're wondering why. So were we. But first we wanted to know how small could he visualize these particles. So I made it easy and brought up a fairly large diatom that was quite recognizable, and this is a cell it's about 50 microns, that's 50,000ths of a millimeter. It's invisible to the naked eye, but you can see it's fairly large because it's got a star shape to it. And sure enough, he could visualize that with his hologram. I hope you'll agree that that is the diatom. And then we did it with a ciliate, which is 20 microns, 20,000ths of a millimeter. And sure enough, we could see it in the hologram. Then we ended up scratching our heads. So what? After many pints of good Scottish beer at local Aberdeen pubs, we eventually realized that what we had here was an underwater microscope. A microscope that could capture all the particles in a very large volume of water down to a resolution of 20,000ths of a millimeter. This is hugely important. Think about trying to do this with any other instrument on the planet. You could take a microscope and you could put it underwater perhaps, and you could then maybe look for particles that are 20,000ths of a millimeter, but you'd be doing it in that much water. The smallest drop you can imagine is what you can look at with a microscope. Here he was able to do it in a tank that was 3 feet long by 2 feet by 2 feet. Phenomenal. This instrument now, we published it in one of the top journals. This was a replay of the hologram showing the spatial distribution of every one of those particles in a very large volume captured instantaneously in a 50,000th of a second. This is published in Limnology and Oceanography, which is the top journal in its field. And that instrument now is being developed commercially in Europe to use as an underwater microscope to look at sinking particles in the ocean in large volumes. So that will come out. But of course, being the nomadic microbiologist that I seem to be, I left London and came to Florida, where I worked at an oceanographic center for about 8 years. This is it. It was beautiful. In many ways, I say to myself, why on earth did I leave? I used to have an office up here that looked straight out over the Atlantic Ocean, and it was a very nice place to work. You could go to work in shorts and a t-shirt. But anyway, I was there for about 8 years, and I did a lot of work on fecal contamination on beaches. That's me in my younger days, looking like I'm doing something important on the beach. But what this was a study that was funded by the Environmental Protection Agency, and they were worried about microbes in the sand. So let's just back up for a moment. Why would we use microbiology to look at water and sand? Well, of course, this is the only way we have to test to see if water is safe to swim in. And what we're concerned about at a bathing beach is whether there is any sewage in it, any sewage that has come from humans. Because swimming around in sewage is not a good idea. It makes you very sick. And so they test the waters of every bathing beach in America, and they do it every 2 weeks. And of course, Florida, for Florida, it's hugely important their beaches are clean, because tourism is the #1 industry in the south of Florida. So this actually got high profile, hence the reason I was in the Miami Herald, because people were very worried that Environmental Protection Agency scientists were starting to question their beaches. Why would we be causing concern? Well, up to this point, and even now, all the water is tested in offshore, just in knee-deep water. So you might be sitting watching someone from the Water Regulation Agency collect water samples, and what they're doing is they're going to test them for fecal organisms, and they just collect them where the water is, just about a few meters out. But someone in the Environmental Protection Agency had a brainwave that, well, what happens in the sand? Because sand is a filter. So maybe if you've got water coming in and it's sieving itself through the sand, that the sand is accumulating high numbers of these organisms, which could make you sick, because kids play in the sand, get it in their eyes and in their ears. And so this was a study that was, for the first time ever, was looking at what's in the sand of a bathing beach rather than the water. And it was a great study. I headed it up for 4 years, multi-million-dollar study, and again, high profile. We actually got thrown off a beach by the lifeguards one day in Fort Lauderdale. They didn't want us on the beach, but that was a foolish thing to do for an Environmental Protection Agency study. The mayor of Fort Lauderdale apologized the next day, and of course we had full rights to go on that beach and test it. Hence the reason it gets into the newspapers a lot. This is a great example of using microbes in a technological sense to test water quality, because the one thing about the media that we use, the agar, for these fecal organisms is it's totally specific for the fecal organism. So this really works well. These do grow up, and these tell you how many nasty bugs are present in the water. And these are called indicator bugs. So you're not actually trying to find the pathogen like salmonella that might be in the water. You're trying to find an organism that is always in sewage. So if you find it, you know there is sewage in the water. And that organism of choice is known as enterococci, and you don't need to go into the details, but we filter it through a filter. You grow it up on the filter surface, it divides, forms colonies, and in this case you can count the colonies, and you know that there are 31 bacteria in 100 milliliters of seawater. That's how we do it. It's very simple. And be impressed, because that seawater, those 100 milliliters of seawater, that much, contains 100 million bacteria of many different kinds. But the only ones that grow are the ones from sewage. This is a wonderful power of selective media. It's good fun, great fun doing it. So you grow them up in the lab, 24 hours later you can count the number of fecal organisms, and then you decide what to do with the beach. A good beach has between 0 and 34. A moderate beach has up to 103, and anything above 104 of these enterococci, the beach is closed. So, think about it. Two things. Firstly, having any present you would think isn't particularly good because it does mean you're swimming around in some diluted sewage. But the risk managers have determined that you won't get sick unless there's more than 104 of them in 100 milliliters. But what's the other scary piece of this if you do some logical thinking? So you're sitting there on the beach and you've been swimming your heart out, and you see someone coming, taking a test tube full of water. 24 hours later, they tell you that the beach is closed. Doesn't do you much good. You've been swimming in the sewage. The problem with this kind of beach testing is it takes 24 hours to close the beach, and by then, of course, everyone's been swimming around not knowing the beach is contaminated. Anyway, next time you hear of a beach closing in California, you'll have an appreciation for how they determined that. We did actually find that sand contains huge numbers of these bacteria. That's the normal amount in water, that's the amount in wet sand, and that's the amount in dry sand up near the top of the beach. Scary stuff. No need to go into detail here. It turns out that these are probably harmless. They are the same bacteria, but what's happening in the sand, they start to reproduce. They like the conditions in moist sand, and so they are giving a misleading indication of how much sewage is there. An indicator must not divide in the natural world, otherwise it's a useless indicator. And so what was happening here is the indicator is actually growing up in the sand and giving a false impression of how contaminated the water is. Our recommendation, which they haven't adopted yet, but you never know, they might get wise and do it, is because there was a huge washout of those misleading bacteria from the sand with the tidal cycle, that if you're in knee-deep water, you're probably sampling harmless ones. Hence the reason you need up to 104 to get sick, because then you're really picking up real sewage. So the solution to us seems self-evident. Tell the water managers to go offshore and sample the water, because sewage comes from pipes offshore. So it would make more sense to go off, away from the effect of the sand, and sample further out. And that's what this is all about. It shows that the drop-off in numbers of these organisms that are meant to indicate sewage is quite dramatic by the time you get to 10 meters offshore. These samples were taken, I can't read it, but probably up to 15 meters, and then dropping off markedly once you get away from the influence of the sand. So that was our recommendation to the Environmental Protection Agency. We're still waiting to see if this is adopted as the new standard for beach quality. Oh, just one more that I got interested in, and somebody here might pick it up. This is very current. This was the last research well, more or less the last research I ever got involved with. And again, it's using microbes in a practical sense. There's incredible concern in the world about invasive species. Invasive species are species of everything, microbe, plant, animal, that are coming to our shores from foreign countries. And they're taking over and they're upsetting our ecosystem. So we're concerned about toxic dinoflagellates coming from, say, Singapore or Hong Kong. Or we're worried about zebra mussels coming from, in the case of the infestation that we have in the Great Lakes, coming from Russia. And they're coming to us usually in the ballast water of ships. So when a ship goes over to Hong Kong and drops its cargo, it has to fill up with water to be able to come back to America. Otherwise, it's unstable and rolls over. And that water is then discharged in American ports. And we think there's about 21 billion tons of ballast water dropped into our coastline every year. And the problem with that ballast water is, of course, it contains organisms which could be invasive. The biggest problem we've ever had is with zebra mussels, which have inhabited the Great Lakes and are now extending down Mississippi, Tennessee, Hudson, and Ohio River basins. These mussels have completely destroyed the ecosystem of the Great Lakes. These are filter feeders. They feed on those algae we talked about earlier. One of those mussels can filter 20 liters of water a day. Think about it. It's a lot coming out of one mussel. And so the Great Lakes right now have actually never looked cleaner. The water is very clear because of zebra mussels, but they've destroyed the fishery. There is no fishing in the Great Lakes anymore because of this invasion. It's also cost billions of dollars in clogging pipes, clogging intake pipes, particularly of cooling towers for electricity power plants, etc. It's $1 billion damage because of this invasion. And really, most of the problems in a tank are microbes, because all of the numerical numbers are either due to viruses, bacteria, protozoa, or algae. Very small numbers of these organisms called macroinvertebrates, doesn't matter, like the mussels in a small ballast tank. Look at the numbers of microbes. So what we're really talking about here is how do we kill all those microbes and other things in a ballast tank before it is discharged. And of course we're particularly worried about organisms like these. This is a dinoflagellate. This is actually the one that causes red tides. And if this comes into our coastlines, it can do immense damage, of course, because of shellfish poisoning. So I still sit in the Coast Guard panel trying to, because it's a Coast Guard problem in America, that we have to find a way to find a treatment system to treat water before it is discharged over the side of a ship. And it's a huge problem because the volumes are so immense. And as I say, the Coast Guard has been challenged with it. If you want to know more about this, look up Coast Guard and what they're doing with ballast water treatment. But they are the ones devising the regulations, how much the treatment system must kill. And at this point, we're trying to find ways to test industry's treatment system, because industry is desperate to show they have a system that works. Whoever gets this right will have their system put on every ship in the world, because this is such a big issue. And we are just waiting to find an industry that can step up to the challenge. There are about 53 companies out there at this point in time that claim they have a system that can do this. But we are having to test those, and we test them according to protocols that people like me have dreamt up. So I'm on that panel of 8 that is designing the testing protocols to make sure a system works. And they would rather do it on a land-based system before it actually gets on a ship. But while I was in Florida, I got quite a lot of money from NOAA, the National Oceanographic and Atmospheric Organization, and that was to test systems that had already been put onto ships, because some of them, particularly cruise ships, were putting them on in actual fact to be compliant with California regulations. So I spent a lot of my time on ships like this. It was good fun, I suppose, except I was usually down in the engine room. In this case, testing this system that a company had come up with. This is the system, all of this, designed to kill those organisms. This actually doesn't work. The company has now gone bankrupt. But they need people to test them, because sometimes they don't work. They now have a test system down in Key West. This is in the Navy Yard down in Key West, where they've got full-sized ballast tanks on land. These huge tanks are about the size of a 5-story building. And so they can hold volumes of water that mimic what's in a ship, and then they can run them through companies' test systems. And that's going on right now with different test systems. And the actual tests they are doing are the ones that I have dreamt up along with 7 other scientists from around America. We have lots of fun meeting and trying to get this right. The point I'm trying to make here is, here's microbiology and someone with experience in microbiology setting policy, having to do it ethically, so that we can really hold these companies to some sort of standard that is ethical, so that we're not just putting on systems that don't work and don't protect the environment. And that's the challenge. And finally, because you're going to get away tons of time, the very last piece of research I did was with an undergraduate student in Fresno, and that undergraduate student did research with me for a year. He published 2 papers in medical journals and was admitted to 5 medical schools. He's now at University of San Diego Medical School, and he got in there largely because of the research he did. when he went to his interviews, he was grilled on his research. And so my hint to you is try and get yourself an undergraduate scholarship experience during your undergraduate years if you're wanting to go on into graduate school. But the work Ivan did was with this amoeba, and it's a somewhat nasty amoeba because it can invade your eye and give rise to amoebic keratitis. That's a condition that some people get from wearing contaminated contact lenses. So if you wear contact lenses, make sure you clean them according to the manufacturer's instructions. You do not want to get amoebic keratitis. I have a lot of physician friends who diagnose it because the patients are suicidal with the pain. This amoeba burrows down into your nerve endings in your cornea. But we did a lot of work trying to understand what conditions lead to those infections. And as I say, Ivan published 2 papers. And I'm going to leave it there because we're running out of time. Good luck, everybody, on your exam. I hope this course has been meaningful in some way. And I can't wait to read the reviews.